Perhaps to move into the discussion session, could we get all the speakers to come up the front? And for those of you who are contemplating asking questions, please move to the microphone so we can see you and we can get the discussion happening. Thank you. And if you have a question, please indicate who you are and which speaker you would like to question. But yeah, just come to the microphones and we will work through these in some sort of order. But I think, Bob, you're probably the first.
Yeah, thank you. My question is to Yusaka Uga from Japan. While explanation, he has concluded that the root growth is associated with the drought tolerance and it relates to the yield finally. But our experience working with the system of rice intensification, I hope that a method of cultivation of the rice, which have become very popular in the recent times, in the last decade, again, it disappeared because of the so many practical problems and also the other related issues, where the root growth has been taken into consideration when we have compared in our experimentation with the normal cultivation of the rice, aerobic situation, and also the system of rice intensification. The water use efficiency was more in the system of rice intensification, but it was not similar across the soil types. That means a genotype which is performing under the soils which are having the more bulk density, and the similar genotype which was shown under the light soils where the porous soil is there, the root growth differences were abnormally different. So while conducting the experimentation, many of the speakers, whether it is a dry crop or is a wet crop, are taking into consideration of a single soil type or sometimes hydroponics, the growth, and concluding the remarks, and also focusing on the QTLs finally. But my suggestion is that while doing this sort of experimentation, and also the more adoption, in the reality in the adoption by the farmers in the open fields, can we experiment taking the soils of the heavy, that means high bulk density, and the medium bulk density soils, and also a root which is the plant which was grown under the hydroponics. When we average these things, I hope that we may likely to get the realistic performance of these genotypes or these technologies, which we are working out with almost with a sophisticated equipment with a high throughput equipments. So my suggestion is that...
Excuse me, can you come to your question, please? Can I ask questioners to please not give us a 10-minute diatribe, but to, if you have some preamble, that's fine, but get to the point of your question a bit more quickly, please.
So a genotype is not expected to perform across the different types of the soils, where we cannot expect the uniform growth of the root system. Thank you.
So my answer is so now a good answer, but actually the QTL aeric variance is affected by in each genetic background and also the environment condition. But Actually, the drought in my case, the drought is stable affects the root growth angle in the several environment conditions and the upland condition and the lowland condition and the high condition and the several artificial conditions. So we should validate those QTL effects in the several environment conditions to apply the breeding material. Yeah, this is my point.
Perhaps Bob, I think you were next, and then Graeme.
Bob Sharp, University of Missouri. I have a question for Michael Barch. I was intrigued when you were overexpressing those ABA receptors in maize, the variety of effects that you got. Unless I missed it, you showed some intriguing differences for leaf growth. I don't think you showed us what happened to root growth. Did you see differences in?
This work was done by my US colleagues. I don't have all the details, but I remember that Sean Cutler is working with us on the same topic, and he continues his research now independent of Syngenta. And so I think I have to refer you to Sean Cutler, but for sure there is some effect on root growth controlled by certain ABA receptors. And I'm not quite sure if Sean has published this. But this is for sure, this you can expect.
Yeah, I agree. You can expect. I was just curious if there were differential effects on roots and leaves, or the ones that showed inhibition of leaf growth also showed a dominant effect on root growth, or were there differences in root ratio?
Okay, so I think in the standard screens, this was not assessed. I think they did not do root washing for all these lines. So at least I'm not aware of the data. Okay. Sorry.
Graeme Parker, Australian National University. My question's to Arun Dhanapal. I enjoyed your talk very much. I wondered, when you examined the genes involved, did a rector show up, a rector that was identified in Arabidopsis and also in wheat as being important for carbon isotope discrimination? I wondered whether it showed up in your soybean study.
The question is whether I referred to the genes in Arabidopsis and wheat, right?
Well, just a particular one, erector.
Gene.
No, I don't think we came across the gene in our analysis. Yeah, thank you.
Sorry, Neil.
Neil Turner from University of Western Australia. My question's mainly a comment, really, to Aude Kupel-Ledhu, and that's in relation to nighttime transpiration. While you can show large differences in nighttime transpiration in your glasshouse experiments, which are well ventilated, I wonder how relevant they are when you get to crop canopies. Now, I understand that would be valuable in vines, but in crop canopies, often at night, even when the stomata are open, the transpiration is in fact quite low because you have very high humidities in the canopy, and the actual transpiration, unless it's, and the wind speed is also low, so that transpiration is often quite low at night. And so I just wonder how relevant it is. And I comment on that because we did some studies in soybean in the field many years ago, and you certainly could show the stomata were open, but it was only important on very hot, windy nights in terms of actual transpiration.
Thank you for your comment. So, yes, actually, I think these results could maybe be relevant for other species, cereal species, in the context that IPCC projects higher VPD during the night for the coming years. So maybe that could be the first thing that may change things. And to be more precise about other species, several recent works on wheat and rice have also evidence that they have also evidence significant levels of transpiration during the night. I'm thinking to the work of Walid Sadok and colleagues on wheat, and there is also a poster here on rice. So maybe these are evidences that could probably be also interesting for other species. But obviously, this has to be further investigated.
Tony Fisher, then the person at the back.
Thank you. My question is for Jana Kochlova. Thank you for a very spirited presentation. I hope I understood it correctly, and I think I've learned something now about the transpiration limitation trait. That's what I'm calling it, that the growing conditions affect the expression of the trait. If you grow under low VPD, you see the trait, you see the differences in plant size. If you grow under high VPD, you don't see it. My question is, for the environments that you are targeting or breeding, which is relevant, the low VPD growth environment or the high VPD growth environment?
That is really excellent question. That is exactly what we are trying to do now. We are trying to see our main population of target environments and characterize what are the prevalent conditions there, so that we can then screen the crops in the same or relevant situation to those which are occurring most likely in those target population of environments. And it is really, really possible because during the year in Hyderabad, you get quite a range of temperatures and humidity. So I think our environments are really relevant to most of the semi-arid tropics. So if we have a data, what I want to say, if we know what is the target population of environment, we have quite a range of choices when to screen our material so that it is really relevant to the conditions which we are targeting. I hope it's answering the question.
Okay, P.S. Zaidi from CIMIT. So my question is to Michael, related to managed stress environment, where you target for 40% yield reduction in managed stress drought trials. So the question is how you keep track with that 40% yield reduction while imposing drought stress, because yield comes, at the harvest. So is there any good handle to keep track 40% yield reduction and make a decision that no, it's enough drought or too much drought?
So I think it's, my work is concentrating on research in climatic change and greenhouses. But what I learned from my colleagues who are running these managed stress site environments, it's really an art. You really need to understand, of course, your soil capacities to store soil. So it's a lot of experience. But what they told me as well is that they, for example, look at leaf wilting. So they target a drought intensity where you get leaf rolling at midday. But if the drought progresses further in intensity and the leaf rolling of the corn leaves would be already visible in the morning, then they would start re-irrigating. So then, and I think, and then with this visual guidance, plus of course they have soil humidity sensors. So I think it's experience measuring soil humidity and looking at leaf rolling. And then considering all these effects, they managed to reduce yield quite consistently by 40%.
Okay, we have a question from a lady in the middle here.
Actually, I don't have any question. I just want some information from Dr. Yusaku Yuga, Japan. I'm Pooja Rai, PhD scholar from BCKV, West Bengal, and rice is the major crop for production and for consumption in West Bengal, India. So I'm trying to find out the new alleles for DRO1 in the land races of West Bengal. And I want to develop allele-specific marker for the region, as I am comparing the coding sequences as well as the promoter sequences also. I wanted information about the promoter region, the upstream sequences of DRO1. So can you please tell us some of the real-time primers for the real-time PCR and all? Or you can send me, mail me, or those, because I'm getting problem in sequencing this.
Yeah, sure. I provide the information. Yeah.
Francois?
It's a question to Jana, but probably also to Arun or then Michael. Yeah, I need just to know whether I understood anything at all. I have transpiration efficiency is a ratio between transpiration and photosynthesis. And obviously one would like to increase one, decrease the other one. I have the impression from all your talks that finally we are better off if we change the timing rather than just the mechanisms of one or the other, the intrinsic mechanisms. That applies obviously to your talk, Shana, because VPD, afternoon versus morning, etc. Obviously applies to day and night, applies probably to Michael. When he applies at a good time. Probably you didn't say that, but probably you apply at a given time of the day. So is this idea stupid, or is it a common ground for all your talks?
Well, thanks so much, Francois, for your talk or for your question. I think I think that life is and the breeding and crop improvement is a matter of priorities, in fact. So what I meant to convey the message is if we don't have solved timing of water extraction, then we can't even talk about the transpiration efficiency. I think that all the crop improvement have to be highly structured the same way how we, for example, parameterize our models for it. So first, if we don't get the canopy, the flowering time right in our agroecologies, there is no talking about timing of the water use and the water use efficiency. If there is, if we settle on the most likely efficient flowering time or fitting flowering time, then only we can start talking about the canopy size. And if we have the canopy size, if we matched with the flowering time, then only we can start talking about how conductive that canopy is, and then fit those nuances within these major issues. And I think Mark was also talking about it. For example, he was saying what I really liked in his talk, that now when they have access to the limited transpiration rate, they can afford to grow the plants which have higher canopy size, which then is a higher sink or source of photosynthesis than all the processes linked to it. So it's really about the pyramiding. So one cannot exist without without the other, I think. So.
And so regarding the question of the timing, so if we look at, for example, this chemical solution to transiently reduce water use, like Vincent Valdez is always showing in his work, that we would also, of course, target a chemical application in the vegetative phase in corn when there is still enough water. And then under well-watered conditions, the plant would start saving water, either due to stomatal control and or leaf growth reduction. And the advantage would be, of course, that if this tool would work later on in the hand of the farmer, that the farmer has a flexible tool to adapt the traits of the plant with the chemical tool. And then, of course, if we would apply such a chemical, we would most likely apply it in the morning so that you have immediately for the rest of the day a maximum effect on the day of application, because also the chemicals will be metabolized and so on. So we don't expect a chemical lasting forever. So to make good use of it, we would apply it then early in the morning.
Maybe just a quick comment to complement. I agree, Francois, and I think that maybe all these evidences converge to suggest that water use efficiency should probably be seen more and more as a really integrative trait in both time and space at the whole plant level, and maybe only working that way we will be able to make more progress that has been limited when focusing on the daytime mechanisms, such as characterizing water use efficiency through gas exchange in the daytime or carbon isotope discrimination. That is only focusing on the coupling between both traits. So maybe a more integrative trait.
If I can take a chairman's prerogative and make a comment on that comment. I think it's an interesting comment, and Ord's remark about TE being an integrative trait I think is also relevant. But perhaps the two things have some degree of independence, and they're not all just about looking at timing of effects. And perhaps the best response to Francois' comment is, it depends. So with that, can I take the question at the back?
Hello, my name is Jamil Khan. I'm from University of Agricultural Science, Bangalore, and my question is to Professor Yoga. And I'm following his research from the publication with Dr. Jagdish Rane on drought QTL. So my question is that drought region is the QTL which probably there was one more paper earlier published on the same region which was RM201. So is there any relationship between that QTL and the QTL which you have found at drought 1? Both are linked with the root rate. And the second part of the question is, what is the possible future for using of that drought 1 in dissecting up to the S&P level? So that will be useful for future breeding line of work.
For the first question, sorry that I don't know that the relationship between your question QTL and the drought 1. And the second question is.
So there was a possible QTL earlier identified. That was RM201 in the same region of the chromosome where drought 1 is identified. So what I'm trying to get the answer is there any relationship between those two QTLs, or those are separate QTLs?
I don't know.
Okay, we can talk about that later. But what about the SNP detection? Single nucleotide SNPs for that region, drought?
I didn't conduct the DNA sequence of the so many rice variety, but yes, drought 1 have a several haplotype in the exon region, but I'm not sure that the other outside of the drought 1 region. So
will it be a wise decision to go for SNP detection in that region?
I'm sorry, I don't know. I'm not data will answer your question.
Thank you.
Do we have any more questions? It looks like we might have exhausted the supply. Oh, there's one over here, Aki.
Akihiko Kamoshita, University of Tokyo. I have two questions. One is to Dr. Uga, or maybe Brian Hammer. In other than rice, how important the regulation of root growth angle in other cereal? You mentioned Medicago, but are there other crops, particularly in cereal, which the root growth angle, regulation of root growth angle will be important for the growth morphology of plant? And the second question is about transpiration efficiency. Maybe first was third speaker. I think in my experience, if water stress, leaf growth is so sensitive. So there is a change in the leaf thickness. Leaf morphology changes. And the transpiration efficiency should be measured under the condition where the leaf thickness change. And under that condition, would you comment how important the changes in the leaf thickness for their transpiration efficiency?
That's the first question.
Yes. I'm not so familiar to the other crops, but Jonathan Lynch reported that steeper roots system are very important to the getting the nitrogen and the water from the soil, and also the sorghum and the wheat. Many researchers reported that the root growth angle is important. Yeah.
Same genetic mechanisms.
But yeah, I'm not sure that same the mechanism, but I think that the drought 1 homolog gene will be associated with a galactic response in the oxygen pathway, I believe.
I can make a quick comment and make an ad to come to Vijaya Singh's talk on Friday, I think, where she will cover this work in sorghum and some of the genetic connections. Question on TE and specifically, Sharon?
So that's really a curious question. Thanks for it. I definitely agree that the leaf thickness is changing if we grow the plants in a different conditions. But I don't think that the leaf thickness is a good indicator of the transpiration efficiency, because what I have seen in pearl millet, and you can visit one of our students who has a poster on this, then their higher water use efficiency is associated with the plant which has thinner leaves, while in chickpea work we are seeing exactly the opposite. Generally the plant which is more water use efficient has actually thicker leaves. So, well, it might be associated, but it's definitely not good proxy for water use efficiency.
Recording is on.
Okay, if we're out of questions, I think it's time to take a break. Are there any announcements that need? We need to have people back by 11:10.
Please thanks our coaches for organizing this wonderful session. Thank you so much, both of